So first up, what I would like to do is to level set our conversation around off-target effects for safer genome editing. And Eric, I'd like to start with you, please. Let's start off by talking about what is an off-target effect, first of all, and what is the significance of these off-target effects when considering the use of CRISPR in both germline and somatic cells? So, Eric, you have the floor.
All right. Thank you, Erin. So I'd like to level set, I think, first of all, on overall, when we're talking about a gene therapy, where, as the audience is well aware, we're talking about modifying or manipulating the expression of a gene to alter a biological property of a cell that's of interest for therapeutic use. Gene editing can be seen as a subset of gene therapy where we're going to focus primarily today, in which CRISPR is one of those tools to edit genes. This could be used to replace a healthy copy of a gene, inactivating a gene, or introducing a new gene for the therapeutic purpose. This can be done in vivo or ex vivo, and along with CRISPR, some tools are zinc finger nucleases, talons, meganucleases, and viral vectors. So that's what we're talking about when we're speaking of gene editing. And when we're talking about an on-target effect, that would be an intended gene edit to have a therapeutic effect. When we're talking about off-target, that's something where we target a different area in the genome than what was intended, right? And so and I think adjacent to this is also, you could have an on-target effect with an unintended biological effect. So really what we're going to be talking about today is how you can, different strategies to reduce off-target effects, how to detect other strategies to control for it. So thank you, Erin.
Yeah, sure. Thank you for that. It always helps to kind of make sure we're all on the same page when we are starting off our event. So that's great information. While I have you, and then we're going to, of course, hear from Steve next on this, I'd like to, if you could provide 1 or 2 points regarding off-target effects, in genome editing, you'd like to share with the audience. So whether that be nuances, challenges, opportunities, that type of thing. To kind of bring us all on the same page.
Yeah, and I realized as I ended my statement there, I didn't touch on the germline versus somatic cells as well, and I think this is important, and we can talk about it more. But real quick, just to touch on that point, I think if you're making a gene edit ex vivo outside the body on a cell, then you don't have to necessarily consider this. But for an in vivo gene edit, then you're targeting your cell type of interest, but there is a risk of targeting the genome. And so off-target effects have a larger risk for in vivo therapies, where you could affect the genome and exponentially affect your risk, as this off-target effect could be passed on to future generations. So it needs to be, it elevates the risk a lot in that scenario. So, okay, and then to move on to the next question. So for what I wanted to touch on, I guess, here was, there was recently guidance from the FDA on gene editing techniques, which I would suggest, looking that up if you haven't seen it already, in January 2024, which built on previous guidance for cell and gene therapies, but this one was specifically for gene therapies and gene editing. And I wanted to touch on a couple topics here I thought was interesting coming out of that. So a lot of it was things that you would normally consider for any drug product, characterizing your identity, sterility, purity of your product, to essentially limit as much of the unknown as possible that you would consider for any drug product. But there were some interesting topics specific for gene therapies, which one was considering all the aspects of your drug product, which includes the carrier to the correct cell type if it's in vivo, as well as all the gene editing components. So for CRISPR, both the Cas9 enzyme, the guide RNAs. So understanding individually as well as cumulatively your drug product as much as possible, which you have to consider more than other drug products. Another I think is interesting is how the preclinical animal model that you're studying relates to your targeted clinical population. So as species have evolved, in some species they duplicated their genomes. We know there's new genes that evolve over time as well, and so considering the similarities and differences there can help to understand how you might have off-target effects as well. One of the key concepts they brought up in the guidance was therapeutic editing threshold. So related to, how much gene editing do you need to succeed in attaining your therapeutic effect. This can relate to the durability of your gene editor. So how long is it around in your cell, and what's the activity of the gene editor as well. And these are really important to consider, trying to match that gene threshold with the therapeutic threshold with those characteristics of your gene editor. And then I think the final one I'll touch on here is some manufacturing considerations when you're thinking about ex vivo therapies is what's the timing within your gene editing step, or what's the timing of your gene editing step within the manufacturing process? And like I mentioned earlier, can you characterize the individual products as well as the overall drug product and do it in a way that's going to be scalable, so you can understand if you're doing preclinical studies versus when you're scaling to much larger patient populations in later clinical trials.
Thanks, Eric. That was really detailed. Appreciate all of that information. And Steve, I'd like to pose the same question to you, and perhaps we're going to cover the guidance more, or whatever you think regarding a few points of off-target effects in genome editing you'd like to share with the audience.
Absolutely, and thank you, Eric, for that introduction. At Caribou, we implement the CRISPR system for our genome editing. And most of what we're doing is either a gene knockout, right, a gene disruption, or a gene knock-in predominantly. And we implement the technology for ex vivo cell therapies, but of course it could be implemented for direct in vivo gene therapy. And we actually had a presentation at ASGCT about a week ago on that technology as well. But in terms of off targets, it really depends upon the tools that you have to evaluate the off targets. And there are many tools that are available in the field. And, they vary. They vary with respect to their capabilities of capturing off targets. And so, the off target in the CRISPR world could mean as many as 5 or 6 mismatches between the guide RNA and the chromosome. And so because of that, you really have to have a tool that can find them. And so it's not necessarily that you want to screen the whole genome deeply to look for them. You want to be able to know where to look and then do your sequencing evaluation where you look. And so, we developed a system many years ago called CiteSeq that uses a biochemical approach where you biochemically identify them by taking purified DNA, put it in a test tube, and add your CRISPR system, your favorite CRISPR system, and evaluate all of the potential off targets. And then you look at those sites that happen, because there may be quite a lot, in cells when you perform it. Or, if you're going to do a mouse experiment, you could look maybe in the liver, depending upon where you're targeting. So that's a way to do deep sequencing analysis to really understand, are you hitting those sites? And one of the things that we've implemented at Caribou, but it is proprietary to the company, is modification of the guide. So guides are naturally all RNA as they come out of the bacterial systems that created them. But we have modified them to introduce DNA residues into the backbone, and that detunes their affinity to some degree. And what that does is it changes it such that if you have a high affinity interaction where you have a perfect match between the guide and the chromosomal target, which would be your on target, you can get a conformational change of the Cas enzyme, either Cas9 or Cas12a. We've done both. And that will lead to an on-target event. The off-targets, though, are limited because you've detuned the affinity so much that if there are mismatches, especially the ones that could happen with an all-RNA guide, we don't detect them within the limits of detection of the assay. And these assays typically are deep sequencing assays where you're looking at maybe 1 in 1,000 reads. And so I think it really boils down to the tools that you have to find them. And so if you're performing an in-cell experiment, you can look for them using a variety of different tools, some of which are described in the guidance from the FDA. And some of them are commercially available. And then some tools you'd have to develop them yourself. And then there's, the other things that we don't look at, right? So I call it the flashlight in the darkness, right? And if you have a flashlight that can find these changes, then you'll find them. Otherwise, you won't see them. So there could be something as simple as the type of mutation that occurs via CRISPR, which happens often in a very small location. So it could be maybe, 20 to 30 nucleotides where there's a modification and then the cell repairs it and you get an insertion or deletion, what we call indels. But there could be huge deletions that occur downstream that you'll miss because you're not looking for them. The other thing that can happen is if you're trying to do a gene insertion instead of a gene knockout, you might be inserting that gene somewhere else in the genome that you didn't intend to. And so you have to be able to have tools to look for these things. So I think that's really the crux of it.